So yeah, thank you all for coming, and thank you for the organizers of this conference to have me here. And I'm Jiawei. I'm a postdoc at Purdue University. I'm working with Dr. Qixing He. So today I'm happy to talk about a newly constructed theoretical framework to predict the strain diversity. We call it MultiSEED, which means multi-strain equal evolutionary dynamics. Yeah, we focus on the strain diversity in human diseases, and we define strains as antigenically different subtypes of the pathogens. So if we see some human diseases, we can observe a lot of different strain diversity levels. For example, for some diseases like measles and chickenpox, we only have one strain. But for some other diseases like influenza, we can have the seasonal replacement of strains. If we see this figure here, we can see these different colors, and we see the replacement of dominant strains over every 4 to 5 years. And we even can observe the circulation of multiple strains at the same time in some other diseases. Like HIV, we see almost 10 subtypes. And in this bacteria pathogen called Strep pneumoniae, people have been detecting over 90 different serotypes. Another very extreme example is malaria. So the malaria parasite is with very high polymorphism because of the antigen part is encoded by a gene family called VAR gene. So each malaria parasite can have up to 60 different VAR genes. And in this network, each node means a malaria parasite, and these nodes are connected together when they share 3 of the VAR genes. But remember that we can have 60 different VAR genes in every parasite. So we can imagine that especially when the transmission intensity is high, we might be able to see different thousands of different strains of malaria parasites. Because we see such a huge difference in strain diversity, we're interested in finding a model framework to predict the strain diversity, and we aim to predict in a long term, so combining both the ecological and the evolutionary timescales. And then we would like to use this framework to get some idea of what factors can determine the strain diversity. So there are some areas of factors that we are thinking of. Interactive factors, which are involving in the pathogen life history and transmission pathways, such as the transmission rate and how fast the diseases can spread, or like how fast the host can get recovered. And intrinsic immunological factors, such as strain-specific immunity or some general protection by cross-immunity. An intrinsic genetic factor means how fast can the pathogens produce new strains. And also we would like to see what factors are in the influence of external forces such as population migration and control measures. Actually, there are some previous models about the dynamics of multiple strains, but why they are not that useful in answering our question. The previous ecological models, they usually do it in the compartmental modeling methods, but they often have limited number of strains, like only 2 or 3. And most of them focus on the short-term invasion process only. For example, in this very recent study, they want to see if 2 strains can mutually invade into each other, but they only focus on the invasion time window. So they find out that in almost every scenario, these strains can mutually invade, but they haven't considered the stochastic processes within that. And also they are not considering any long-term evolutionary processes, so they are not that useful in our question. And on the other side, previous evolutionary models, they are trying to track the dynamics of different strains, but the methods they usually use are the individual-based models, and they run some stochastic simulations. They are good, but then they are restricted under very specific parameter combinations. And also they are restricted by the computing speed. So these factors are limiting the analytical investigation over a very large parameter space. So we try to resolve these drawbacks by constructing our framework called MultiSEED, which is a combination of a status-based multi-strain model, and also we consider continuous time stochastic processes, so we can predict the strain diversity over the long term, and we can test it over a large number of parameters and a large parameter space. I will start with this model structure. Like, it is actually a status-based model, but it is derived from the very basic and classic SIRS model in infectious diseases. So here we see this Xi means the susceptible host to strain I, and we are assuming multiple strains and they are having competition. And this X nothing means the naive hosts with no previous infection history. And this part, it is meaning that the hosts that are susceptible to strain I, but with some previous infections. Here we have Y as the infected class and ZI as the recovered class. So these are the explanation of these classes. And so what are the differences in transmission? So we consider the cross immunity here. If the hosts are with some certain transmission or infection history, then they can have a reduced susceptibility here indicated by rho. And otherwise, we have the adjusted R0, the basic reproduction number, to allow for the mixed infections and the strain competitions within hosts. So we would also have like the recovery here and also the waning immunity of strain-specific strain-specific immunity. And these are some explanations for the parameters. So we also have demographic factors like host birth and death. And then I would like to introduce this parameter first. We call it relative resource recruitment rate, and it is equal to epsilon1 plus epsilon2. So why is that? Let's think about how the pathogens can get resources from hosts. So it can be either through the immunity loss of the strain-specific immunity, and also the birth of new susceptible hosts. But we are also rescaling it by considering the parasite death, either through recovery of infected hosts or the death of infected hosts. So next, I will move to the continuous time stochastic processes. So we would like to find out the strain dynamics. So here on the left part, we have the ecological timescale, and on the right side, we are having the evolutionary timescale. And the x-axis is just the flow of time. Y-axis is the number of infections. So we consider that in each case, if we think about a newly emerged strain in the system, at first, it might only cause very few infections. So it might randomly go extinct very fast. So we call this a fizzle phase. Then after it overcomes the fizzle phase, it will get to its first epidemic by having the infection peak. But that's the number of infection will sharply decrease because of the decreases in susceptible and infected hosts. So we call it epidemic burnout. Only if one new strain overcomes the fizzle stage and the burnout, then it will become successfully invading into the system. So then it moves from the class of transient strains to persisting strains. For the persisting strains, they might also have the epidemic burnout because of the new strain invasion, and I will show in the next slide. And also we use the OU process, the diffusion approximation, to estimate the long-term extinction rate of the persisting strains. So with all these processes and these rates, we can get to an equilibrium status where the number of strains in the transient class and in the persisting class, they both do not change. So for that N1 plus N2, we define it as the long-term strain diversity. So now I will show the burnout. We consider the red one as an invading strain and the blue one as a resident strain. So it is during the invasion of the red strain. So we can see that in this area, we run some simulations, and both the red strain and the blue strain can go extinct sometimes when their fraction infected is dropping down below the equilibrium value. So we can use some published theorem called Kendall's Q to get the probability of burnout during this time window. So then we can write out another set of equations for the dynamics of the transient strains and the persisting strains. So here, this part, it means the innovation rate of new strains, and then P1W is the probability of escaping the fizzle stage. TE is the burnout time during the invasion process in this time window, and it can be calculated by doing some integrations over this time window and also use the theorem of Kendall's Q. So these KQI and KQE are the probability of burnout for the invading strains and for the existing resident strains. So by solving this set of differential equations, we can get the equilibrium values of N1 and N2. So before I talk about some conclusions we get from our framework, let's first do the validification. So we do this by comparing the N1 and N2 from MultiSEED to the simulation of agent-based model. So we have four specific parameter spaces for these 4 human diseases. We put them in the 2 systems and we compare the number of transient and persisting strains. If we see the red color, we see that the transient strains from these 2 systems are roughly matching to each other, which is great. But for the blue color, the 3 diseases are fine. For HIV, it is not that great. We think it might be because of the relatively small host population size of HIV, because of it is dependent on human behavior. So that in this case, maybe using a diffusion approximation is not that perfect. But these results kind of verify that our framework can get to reasonable strain diversity over the long term. So now let's think about what factors can affect influence the strain diversity. So we are having these parameters. Epsilon, the resource recruitment rate I just introduced. R0 is the pathogen basic reproduction number, which means the number of secondary infections from one infected host in a totally susceptible host population. NH is the host population, not on the level of consensus, but the host population size which are reachable for the pathogens as resources. Nu is the strain innovation rate, how fast the pathogen can get to new strains. And then Rho is the reduced susceptibility under the protection of cross-immunity. So the first 3 parameters are kind of more involved in the life history and transmission pathway, while of course, Nu is the intrinsic genetic factor of the pathogen, and then Epsilon and the Rho are kind of related to immunological factors, because in Epsilon we are also including the waning of strain-specific immunity. And then these first 3 parameters are also under larger influence by external factors such as migration, control measures, or global change. So let's see how these parameters can affect the strain diversity. So first, I fix these 3 parameters and I see the interplay of resource recruitment rate and transmissibility represented by the basic reproduction number. It is a very interesting nonlinear pattern, but we see the different changes of colors here. So in the bottom left part, we see the dark blue color, which means strain diversity is 0. We cannot see the accumulation of strains over the long term. It can match acute infections or transient epidemics where the strains get burned out, sometimes seasonal dynamics and the zoonotic spillover. So let's move to the top right part, and where we see strain diversity is larger than 0, we might be able to see the mixed infections of different strains. But in fact, in reality, this part is seldom observed with both large epsilon and the large R0. Then I will look at the effects of other parameters. So by default, we are having these parameter values, but on the left, I'm changing the magnitude of the host population size. And on the right part, I'm changing the magnitude of the strain innovation rate. So let's first look at these lines, which are representing the theoretical equilibrium value of strain diversity that we obtain from MultiSEED. It is a little bit counterintuitive that actually the host population size is having larger effects on the long-term strain diversity. But not all diseases are coexisting with human for that long enough time. So some of them, maybe they are not yet reaching that equilibrium status. So we also see the influence of limited time by running some Markov processes with a restricted number of generations. Then we compare these crosses and shades with the lines. We can see that strain innovation rate can have more influence on strain diversity under the limited coevolutionary time of hosts and pathogens, but still the effect is not as large as host population size. I'm not showing the effect of the cross immunity like Rho here, because the effect is even more minor compared to the strain innovation rate. So in conclusion, we think that these 3 parameters are having larger influence on the long-term strain diversity, and they are more kind of belonging to the interactive factors rather than the immunological factors. And like Nu and Rho, they are having less influence on the strain diversity. And the first 3 are also under more influence of external factors like migration and control measures. And we also would like to find out what we can get, whether we can get some ideas of the regimes of strain dynamics by applying MultiSEED to the parameter space of these 4 human diseases indicated by the light blue rectangles here. So what are the regimes of strain dynamics? We may be able to see 3 different regimes. Like extinction, it means there will be no long-term accumulation of strains, and the replacement, it means that over certain time period, we can see the replacement of dominant strains. And coexistence, it means the strains can exist for a long time, and then they are kind of stable in the system. So if we see the bottom figure, we want to show that MultiSEED can reproduce the replacement in influenza A and HIV-1 with relatively small numbers of strains. And for pneumococcus, which is caused by that bacteria called Strep pneumoniae, and for malaria, we can reproduce the long-term coexistence of a relatively large number of strains. Yeah, so there are some take-home messages. So we have this theoretical framework, which is combining deterministic models and continuous time stochastic processes. And we validify our model by comparing to the simulations of agent-based models. And we find out that the maintenance of strain diversity is more dependent on the interactive factors and external forces, rather than the genetic and immunological factors. And finally, we show that MultiSEED can reproduce the regimes of strain dynamics in several human diseases. And in the future, we want to find out to test this framework in other systems, such as plant or animal systems, to see if it can apply. So this work is still ongoing, and we are kind of writing. So sorry, I cannot give a link for the bioarchive, but hopefully, I think we can finish it very soon. Yeah, so thank you for all listening. I'm happy to take any questions. And if you have further questions, you can reach out to me and Qixing, who is my advisor. So yeah, thank you for listening.
We have time for questions, and Puneet, can you get started?
Very cool talk. I'm a little bit curious about how you're doing like mutations or innovations. What parameters are those leaving constant? What parameters are they changing? And over long times, are like invading strains getting better and better? Or is it kind of like a self-similar like Red Queen thing?
So for the mutation, we are just we find out the mutation rates or recombinations from some literatures, and then we just also find out like the infections, like the generation time by like the incubation period, or like how long the infections take. So we kind of rescale the mutation rate to be like for each infection generation, and then we are allowing it to, like, it might happen for all the time. But yeah, I forgot to mention that. So in the model right now, for simplification, that we are assuming that each strain is equivalent to each other. So we are not having that heterogeneity in the strains. So right now we don't consider that, but maybe in the future we should add that part. Yeah, so I hope it answers part of your question.
All right, so the only difference between strains is like immune escape, basically. That's the sort of...
Yeah, basically, yeah, depending on like the immunity.
Thanks.
Great, let's thank Jiawei again.